This UCSD TV program is a presentation of University of California Television for educational and non-commercial use only.
I think we'll move on to the next talk, talking about a couple of extinct genomes, the Neanderthals and the Denisovans.
All right, so I'm Ed Green from University of California, Santa Cruz, and I'm going to talk to you about the genomes of our closest extinct relatives, the Neanderthal and Denisovans. So in asking this question about humanness and human uniqueness, we tend to define this as how we are different from the things that are closest to us. And our closest living relatives, as we heard, are the other great apes, the chimpanzee, bonobo, the orangutan, and the gorilla. And this is a useful way to define human uniqueness because there are these things that are evolutionarily pretty close to us. The chimp and bonobo, as we've heard, are about 6 million years diverged. If we go back in time 6 million years, there's no chimpanzee and there's no bonobo and there's no human. There's only the common ancestor of these two. And in the time since then, the last 6 million years, we have become different from this common ancestor as the chimpanzee has become different from the common ancestor. And that's one way of saying how we are unique. What are these last few changes that happen since our common ancestor with chimp? We can carefully observe chimps, look at them and look at humans and make long lists and interesting lists of things that differ between humans and our closest living relatives. And these lists then are in some sense an answer to this question. What is it that is unique about humans? How are we different? Because every human individual starts with this genome, which resides on chromosomes that you get from your mother and your father. Everybody in the room started life as a single fertilized egg with largely just the information that's encoded on our chromosomes. And it specifies these human differences. Every human is completely heritable for these human characteristics, and likewise for the chimpanzee. We would really like to know how this genetic information encodes these differences. What is it that differences between humans and chimps, what is this map between these genetic differences and the biological differences that we might care about? We can rephrase this question, how are we unique, and ask instead what makes us unique genetically. And it's a good time to ask this question because we have lots of genomes from the great apes. Soon we'll have all of those, as we just heard, and get something like a satisfying answer of all of the specific places where we differ from the things that are most similar to us. There are about 35 million places in our genome where we're different than a chimpanzee, and a few million insertions and deletions. 9 of our chromosomes have undergone these inversions, and our chromosome number 2 is the result of a fusion event. This second biggest chromosome exists as 2 discrete chromosomes in the other great apes. So this then is kind of the playing field for defining the genetic basis of human uniqueness. We've got these millions of things to look at genetically and long lists of biological and behavioral and physiological differences, and we'd like to make a map. But that's a hard problem. There are many millions of these things, and understanding the biology behind them, how the genetic information goes through all the steps that Ajit talked about, through gene expression and protein expression and interacting with the environment to make the differences between humans and chimpanzees. So is there any way to make this easier? Is there anything that's closer? Well, there is, and I'll tell you about that in a second. But first, for this audience, I thought it might be fun just to do this thought experiment. We know that, as we just heard, orangutans are critically endangered. Their numbers are dwindling due to habitat loss. This is true for all of the great apes, except for us. The great ape that likes to do math and write poetry is doing great. It's kind of a revenge of the nerds story where we are taking over the world and all these other guys who don't do that are finding the place where they live going away. It took some time for us to have this scientific enlightenment to formulate a model of evolutionary history where we put ourselves in the proper place. This is despite the fact that there are these great ape species that are not so distantly related to us. If it had been the case instead that we reached this enlightenment and started really making progress with this after these guys were gone and we're left instead only with our closest relatives being things with funny faces and tails, it might have been an even harder thing for us to do as a species. Okay, so things that are closer are all dead, they're all extinct, but we find lots of their bones and we argue about how they are related to one another. This hominid fossil record is a fairly contentious thing. The broad strokes of this, I think, are pretty well settled. And one thing that is settled, our closest extinct relatives are these guys here, the Neanderthals. They are morphologically distinct group from humans, even from the modern humans that were contemporaneous with Neanderthals. So here is the cranium of a Cro-Magnon, the modern human, anatomically modern human, and from a classical Neanderthal. And if you look closely, you can notice some differences. There's this brow ridge and an occipital bun, and the brain case is lower and longer and slung back. And postcranially, there are some other differences, but these focusing on the differences here kind of obscures the big picture story, which is that they're very similar to us morphologically in the context of the next closest thing that is still living, which is the chimpanzee. So Neanderthals and humans are morphologically very similar, and it turns out they're genetically very similar too. Temporally, Neanderthals show up in the fossil record about 200,000 years ago, and then they disappear about 30,000 years ago. Our species shows up about 130,000 years ago, and of course we're still alive today. So for most of the time that we have been around, and for most of the time that Neanderthals were around, we shared the planet together. It's only a rather recent development that we are the only hominin that walks around the world. So we overlapped in time. Neanderthals geographically are mainly known in Europe, but their range we know now extended into the Middle East and into Asia. And so they would be a very useful thing to study genetically, and it turns out that this is possible. Before I start talking about that, I'm going to do a little bit of the Ajit-style kindergarten genetics, and it turns out that this is very necessary for explaining the genetic comparison between humans and Neanderthals, because unlike comparing us with other great apes or other species, the genetic differences between humans and Neanderthals are largely not fixed differences. We talk about a human genome sequence and the orangutan genome sequence or the chimpanzee genome sequence. Of course, there are differences between humans and between orangutans and between chimpanzees. And the difference between human and Neanderthal is largely the difference between humans who are alive today. And I'll explain why that's the case in a minute. But the point here of this slide is that kind of explaining inheritance in diploid sexually reproducing species like us. We get our genetic complement, our genome, from our dad and our mom. And our dad has 2 chromosomes, and our mom has 2 chromosomes that they got from both of their parents. If we imagine that these 2 chromosomes are just these intact bars here, the chromosome that comes from our dad was built just for us. It didn't exist in our dad at all in this way. Instead, it's a recombined version of the chromosome that your dad got from his dad and that your dad got from his mom. And after this recombination, you get this genetic material from your dad, and likewise the same thing, this recombination happens in your mom. So if you look, if we say that the male is on top here, this part of your genome here came only from your dad's mom, not from your dad's dad. That bit of this chromosome is not represented by your dad's dad anywhere in your genome. whereas this part is all your dad's dad. So this recombination that happens through every generation means that we have our own particular unique genealogy if we go back in time. At some place in our genome, we may have our dad's mom's mom's chromosome bit, or our dad's mom's dad's bit at that chromosome. And it turns out that you can reconstruct genealogies of any particular haplotype, any particular region in our genome amongst all people who are alive today. And this, in some sense, must be true. There must be this genealogy that unites everyone. You can take any two people in this room, find the haplotype at some place in their genome, and find the time that they had a common ancestor at that place in their genome. This must be true mathematically because we know that at every level of our own personal genealogy, we have a factor of 2 more ancestors. So 10 generations ago, you have about 1,000 ancestors. 20 generations ago, you have about 1 million. 30 generations ago, you have about 1 billion. Well, 30 generations ago, if we say 30 years per generation, this is in the Middle Ages. This is about year 1100. You have 1 billion ancestors. There weren't 1 billion people on the planet at that time. Even if every single person was an ancestor of you, there just aren't enough people so that it's different. So you coalesce with every other person that you might imagine on the planet at some place in your genome. Okay, so if we imagine now these trees that we have put together, looking at genetic diversity that's alive today, on average, humans coalesce. If you just take a random part of the genome and take 2 random people, 2 humans will coalesce. They'll have a common ancestor for that part of their genome about 450,000 years ago, okay? This is interesting with respect to Neanderthals because the population splits that would become Neanderthals and would become humans happened about 300,000 years ago. Okay? So if we could go back in time 300,000 years ago, there are no humans and there are no Neanderthals. There's the common ancestor of this. Maybe we call this Homo erectus or Homo heidelbergensis. But that population that lived at this time, it harbored genetic diversity. There wasn't just one individual, there were multiple individuals and they had their own differences in their genomes. And the diversity that was present then is largely the diversity that's present still today and was still present within Neanderthals until they went extinct. So at any particular place in your genome, you may be more closely related to a Neanderthal than you are to some other human. And in a different place in your genome, you may be more closely related to a human than you are to a Neanderthal. It just is the stochastic nature of this coalescent process and the fact that our gene trees are deep enough to include Neanderthals. Okay, so it's very useful then, it's very interesting to look at the genomes of Neanderthal because this is, in a way, just a deeply diverging human population that has genetic variation that's still alive today within us. Okay, so Neanderthals sequencing DNA from things that have been extinct falls in this field called ancient DNA, which got its start back in the mid-'80s when DNA was sequenced from the first extinct organism, the quagga. And over the years, more and more extinct organisms had DNA extracted and sequenced from it. These early days in ancient DNA were mostly using a technique called PCR, where you would take DNA out of a bone and try to amplify some intact fragment, and either it was there or it wasn't, and you would get some small sequence and try to make some sense out of this. 97, this was done for the first time in a hominid, the Neanderthal. This field has been revolutionized by this next generation sequencing that Elaine talked about that's making everything cheaper. Next generation sequencing was not invented for ancient DNA, but it may as well have been. All of the weaknesses of next generation sequencing, the short read lengths, for example, they don't affect ancient DNA because the fragments that we get are usually so small that a longer read length wouldn't matter anyway. Okay, so the field has really moved toward this direct high throughput sequencing, next generation sequencing, and it's enabled genome scale projects of extinct organisms, not just looking at one or a few targets at a time. Here's what a few of the machines look like. What we have done is generate about 1 gigabase of data from these 3 bones, which are dated to about 40,000 years ago and came out of this cave in Croatia called Vendia Cave. And One of the things that we have seen from just doing massive amounts of sequencing from all of the DNA that comes out of bones is that very little of it, if we take all of this sequence data and align it to everything in databases of all sequence that's been done thus far, mostly what we see are things unlike anything we've ever seen before. If it does look like something in the databases, usually it's a soil-living bacteria, and then just a few % is Neanderthal. So mostly what the DNA that's in this bone is DNA from microbes that have colonized the bone in the 40,000 years that it's been sitting in the ground. But we focus on this few % and have to sequence a lot to get up to genome scale coverage of the Neanderthal. As I said, we did a little over a gigabase from each of these 3 bones, a smattering of sequence from these other bones that didn't have as much DNA preserved, and now have a little over 1-fold sequence coverage. Okay, so one very difficult thing about doing analysis of Neanderthal DNA, because they fall within the variation of modern humans, it's impossible for most of these small fragments, which are about 60 nucleotides long on average, it's impossible to look at this sequence and say that's Neanderthal or that's human, because it's so similar. Most of these fragments will have no difference from human, except for the 2 or 3% of sequencing error that we get because it's ancient DNA. So to say for sure that it's really Neanderthal DNA, it turns out to be a very difficult thing. We did 3 different approaches to measuring the rate of human contamination. One was to look at the mitochondria. As Ajit mentioned, the mitochondria is maternally inherited. You have only one haplotype. You don't get it from mom and dad. You only get it from mom. So we don't have any heterozygosity to deal with there. We have complete mitochondrial sequence from these Neanderthals, and we identified 135 places in the genomes of this Neanderthal and all modern humans where every human looks different than this Neanderthal. So then every fragment that overlaps one of these positions, we can say, does it have the Neanderthal base or does it have the human base? And from counting this, we get about 27,000 with the Neanderthal base, 73 with the human, and see that's less than 1% contamination. Likewise, we made this assay looking at the Y chromosome. These 3 bones happen to come from female Neanderthals. Females don't have Y chromosome, so the presence of any Y chromosome sequence is by definition then contamination. We saw 4 fragments that were unambiguously on the Y chromosome, and from this we can estimate less than 1% contamination. Another assay we did finally, looking in the nuclear genome, the autosomes, was comparing the genomes of 5 humans that we sequenced and look for places where 2 of the 3 Neanderthal bones, just looking at 2 of them, they differed from all 5 of these humans. So all 5 humans had one base, and 2 of the 3 Neanderthals had a different base. In fact, they had the ancestral chimp base, and these are likely things that happen in humans early on, and now all humans differ from all Neanderthals. And then, once we identified these positions in 2 Neanderthals, we looked in the 3rd one and asked, in this 3rd Neanderthal, is it like the other 2 Neanderthals, or is it like the human contamination? And did this in a round-robin approach where you test each of the 3 Neanderthal bones, and from this we can estimate also less than 1% contamination. So in this way, we bootstrapped ourselves into the Neanderthal genome. We didn't know what it was initially, and it's hard to imagine how you're going to know if it's Neanderthal sequence or not, given that you don't know any Neanderthal sequence, but these are the methods we came up with. Okay, so then comparing human, chimp, and Neanderthal sequence, aligning the 3 of them, we can ask how many differences are specific to the chimpanzee? How many places does the chimp have a different base, or the Neanderthal, or the human? The Neanderthal, if we look at the bases where Neanderthal is different, what we see is there's this huge excess of C to T and G to A. This is characteristic of ancient DNA. There's a deamination process of cytosine. It gets chemically damaged, and then it occurs to us as Cs. It also causes the Gs to be read as As. This is not real evolutionary change. These are not changes that happen during Neanderthal evolution. They're chemical damage that happen in the DNA of this individual Neanderthal after it died. And also, we see a very high rate of all differences in the Neanderthal genome that we have, which is low coverage, single pass sequence reads of this Neanderthal individuals, and we're comparing it to finished genome of human and chimpanzee, which has a very low sequencing error rate. So what we're seeing basically here, if there's a position where the human and the chimp say one thing and Neanderthal says something else, it's almost always error. the vast majority of those positions are just sequencing error. So saying something about Neanderthal-specific changes is very difficult to do right now, despite the fact that we have about one-fold coverage of the Neanderthal genome. The errors accumulate on this Neanderthal branch. However, this branch here is very interesting. These are places where the Neanderthal and the chimp have the same base, and humans are different. This lets us say, what were the changes that happened recently in humans, very recently, since we split off even from our closest extinct relatives, the Neanderthals? And if we just ask, what is the fraction of positions that look like this versus look like this, we can estimate how long ago, on average, Neanderthals and humans diverged from one another. And it turns out that this is about 12.7% of the way back to this common ancestor. If you take your favorite time for this, 6.5 million years or so, that means this is about 800,000 years. So the average genome coalescent between humans and Neanderthals is about 800,000 years ago. Okay, so we also, because we coexisted for a long time, there was this open question forever whether there's any genetic relatedness between humans and Neanderthals. We know we lived at the same time and overlapped geographically even. So to address this, there was this analysis devised by David Reich and Nick Patterson and Jim Mulliken, where we took the genomes from 5 humans that we sequenced who were from around the world, one person from France, one person from China, someone from Papua New Guinea, someone from South Africa, and someone from West Africa, and took all pairwise combinations, all 2 combinations of each of these, the Yorubas from West Africa and the French guy, and find places where they differ, where there's some allelic difference between these 2 people. And then ask, what does the Neanderthal have? Does it match the person from France or the person from Africa? If Neanderthals are a clean outgroup to all humans, if Neanderthals split off from humans before there was any Yoruban or French or Papua New Guinea, and if all of that was later, then Neanderthals should match these 2 equally often. It should be 50-50. But it turns out, so here are the 5 humans that we sequenced, it turns out that it's not 50-50. It is 50-50 if you compare the 2 Africans against each other. Neanderthal is equally similar to San as to Yoruba. It's also equally similar to any of the non-Africans that we compared. Any pairwise combination of the 3 non-Africans. It looks just as much like a French guy as it does someone from Papua New Guinea, or Papua New Guinea as a Han Chinese. These are all statistically indistinguishable from 50-50. However, any combination between African and non-African, there was a small but very statistically significant excess matching between Neanderthal and the non-African. There's a closer genetic relationship between Neanderthals and all non-Africans than Africans. And this is interesting because this is the case even for the Papuan sample, even someone from Papua New Guinea, where Neanderthals are never known to have been, and almost certainly never were. We still see this signal of admixture in people who live there today from Neanderthals. So this is what that analysis looks like. These 2 are similar to each other. Any of these 3 are similar to each other, but any comparison between the 2, there's more relationship between the non-African. So this is the model that we have to explain it. 1 to 4% of the genetic ancestry of any non-African now can be attributed, traced back to Neanderthal. Probably this admixture event happened very early on when humans migrated out of Africa and first came into the Neanderthal range. They picked up this Neanderthal genetic component and then took it with them as they went over the rest of the world. This probably small population then expanded, so we see this in roughly equal proportions all over the world. Okay, so that's the Neanderthal genome. The Denisova is another sample that we found recently that has a very deeply diverging mitochondria sequence. Humans and Neanderthals are sister groups with respect to Denisova in the mitochondria. Not true in the nuclear genome. The Denisova versus the Neanderthal is about the same divergence, which is kind of odd. They appear to be a sister group, and in fact, if you look at allelic differences, Denisova is a sister group with Neanderthals in the nuclear genome compared to humans. So they're a very deeply diverging population that's more distinct from Neanderthals than any 2 populations who are alive today are, but they're more closely related to Neanderthals than they are to humans. We did this same admixture analysis and shockingly found that there is a very strong signal of excess similarity between the Denisova Denisovan and people who today live in Papua New Guinea and not in these other places, a wider sample for this analysis. So in the last 30 seconds, I would like to tell you another application of the Neanderthal data, and really the reason that we started this project in the first place, and that is the genealogy of humans versus Neanderthals across most of the genome looks like this. But what we would like to do is find the places where it looks like this, where there's some adaptive mutation that happened in humans since we split from Neanderthals and was selected, conferred some advantage, so it was selected and is now fixed in humans, places where all humans are more related to each other than they are to Neanderthals. And graphically that looks like this. Sometime in the past we got the mutation that let us do anything that's human unique with respect to Neanderthals, and we see this in all humans now. So that screen is something we're implementing and running now to find the genetic basis of uniqueness that separates even humans from our closest extinct relatives, the Neanderthals. These are some of the key people who worked on this project. The real visionary behind this, invented the field of ancient DNA and has been driving this project for decades, is my former boss, Svante Pabo, and these other guys are very important people who worked on various aspects of this. And if you're ever in beautiful Santa Cruz, come by and see me. Thanks.